Good morning, everybody. Thanks for joining here in person and to our colleagues online. My name is Gerhard Wingender. I'm a research group leader at the Izmir Biomedicine Genome Center, and today's seminar is brought to you in joint collaboration between the project of Chiron and Brayer Boost. For those, if you have questions, please use this QR code to ask your questions online. If you put them in the chat, I will not see them. So one of our partners from Chiron, Philip Grapes from the University of Bern, will present next week a seminar about inflammation and cancer microenvironment. And so we thought it would be a good idea to give you kind of crash course to immunology to get you some ideas to understand maybe a little bit better what Philip Grapes will then talk about next week. So that's what I will try to do, to give you a crash course to immunology. If people hear that I'm an immunologist, people usually ask me, so why do we get sick? And I do actually think that's the wrong question. But before I get to that, I wanted to remind you of something. Bacteria everywhere, right? You know that. Whatever you touch, whatever you eat, it's covered with bacteria. I don't say that to scare you, although you should wash your hands. So the point for me is rather, it's not only that they're all around us, they're also with us. So if you look at our body, we are covered and we're filled with bacteria. The highest concentration of bacteria in nature is actually in the guts of animals, like us. And right, a third of the feces is bacteria. And it's not only that there are a lot, they're also very diverse. So if you just look at the bacteria you have on your skin, if you scratch yourself behind your ear or your belly button, you will disturb an entirely different ecosystem. And the bacteria you have with you are so unique to you as your fingerprints. So a lot, a huge variety. And of course, as you remember, bacteria are really good at multiplying. So I didn't crunch the number on that one. I hope it's roughly correct, but at least we all know that bacteria will always outcompete us in numbers. So, and that's only bacteria, right? There's so many other things which can potentially make us sick. Viruses, fungi, protozoa, helminths, arthropods, all of them can make us sick. So in the end, I think the question is not why do we get sick, but rather, why are we still alive? And of course, the title of my presentation gives it away, right? But what happens if you do not have an immune system? And that's the example of David Vetter, which was unfortunately born with a severe immune deficiency. And he could not handle the world we live in. He had to live in a bubble, sterile air. Everything he ate, everything he drank had to be sterilized. So the immune system keeps you alive. So what is the immune system? Roughly divided into an innate part and an adaptive part. And I will explain why we divide it, because these two parts of the immune system have very different strategies, features, and a different goal. And to remember all these different immune cells, I think the easiest is if you keep remembering that the T cells and the B cells are adaptive. If you remember that, you're fine. So how do they look like? You have here T cells and B cells chilling around in the lymph node. So everything which is black is filled with cells. It's just that it's not labeled. And they're doing their business daily in, day out, walking around, looking for something interesting. So T and B cells, what are those? The T cells represent the cell-mediated immunity. And that is mostly as a contrast to B cells, which represent the humoral immunity, because they make the antibodies. The antibodies are coming to that in a second. But the key defining features of adaptive immune cells, T and B cells, is that they have a T cell receptor for T cells and a B cell receptor for B cells. Not very fancy naming, but at least makes it easy to remember. So these B cells can recognize the antigen directly. And once they recognize it, they can make the B cell receptor in a soluble form, and that is the antibodies. So antibodies are the B cell receptor in a soluble form. T cells cannot recognize it directly. They require some kind of molecule presenting it to it. We come back to that later. But what I wanted to say here is that if you can remember that if the name of the immune cell has a big T or a big B in the name, you already know it's an adaptive immune cell. So you can classify all immune cells right away. So you're 5 minutes in, and I hope you learned already something. So what are the features? So we now know the cells. What are the features of the innate and the adaptive immune cells? So one thing is that the innate immune system is fast. If you scratch your skin, it will become red within a few minutes. The speed of that indicates it is an innate response. So how does it look? Just looking at some immune cells. Here in green, you have neutrophils. So half of the immune cells in your blood are neutrophils. The blue one here is a blood vessel. And here in the tissue, there are some pathogens, in this case, parasites. And you can see how the neutrophils within less than an hour leave the blood and swarm to the parasite. So the plaque again, it's not empty, it's filled with cells, and that's why the cells here seem to be elongated. They need to squeeze away through the tissue to get to the parasite. So that happens fast, right? Within an hour, the innate immune cells reach the target and protect you. In contrast, adaptive immune cells are slow, really slow compared to that. So without going into details of what these curves mean, just think about these two represent the innate immune response. This one represents the adaptive immune response. And you can see here in time scale in days that it takes several days before the adaptive immune response kicks in. But the virus is already controlled by that time, right? So the adaptive immune system joins the party when the party is basically over. So they're really slow. And that has to do with the way the immune system is recognizing the pathogen. In the case of innate immune cells, they have a low diversity of the recognition. In the case of the adaptive immune cells, it's a very high diversity. And that is important to really understand these two arms of the immune system. So I want to expand on that a little bit. So what is meant with the recognition diversity? So in the case of the innate receptors, those which are expressed mostly by the innate immune cells, we have about a few dozens of those. And they are shared by not all the immune cells, but all humans. So again, if you take a drop of blood, take a neutrophil, hundreds of thousands within the drop of blood, and you look at them, they look all the same. They will use the same receptors. If you look at the drop of blood of you, the neutrophils will use the same receptors. Any human on the planet will use the same receptors. In contrast, the T cell receptor and the B cell receptor is extreme variable. Billions within your own body. If you look at all the T cells in your body and look at those which have never seen an antigen before, you might have a handful of T cells with the same T cell receptor. And where does it come from? So these innate receptors have been first described by their similarities in humans to Drosophila, like 600 million years of evolution, and the receptors were still similar, which means that they're highly conserved, they're encoded in the genome, you inherit them as they are from your parents. And that goes back millions of years ago. In contrast, the T and B cell receptor is produced every time in your life through an almost random process. And this random process means that even if you have an identical twin, the T cells and B cells of your twin will be different than yours. And because it's random, it's not clear beforehand what it would recognize. What does it mean? In the case of the innate receptors, these recognize structures which have been tested by evolution. Right? Drosophila have a receptor which is very similar to one we have. if something worked 600 million years ago, it probably will work tomorrow for you. In contrast, the adaptive immune cells make a receptor, which is an unknown specificity. So they recognize the unknown. So the innate immune cells recognize what worked in the past. The adaptive immune cells try to recognize what might happen tomorrow. And these defining features make the distinction between the innate and the adaptive immune system. And these receptors, this distinction is directly linked to the last main feature of this immune system, and that is memory. So you scratch your skin, it becomes red within a few minutes. You scratch your skin again next week, next year, in 10 years, it will be the same response. The innate immune system does not need to remember what happened during your lifetime. It has a memory of 600 million years of evolution. In contrast, every single vaccine you ever received is aiming to educate your adaptive immune cells. Vaccines have been one of the biggest inventions, of course, of humanity, saving hundreds of millions of lives. And just to give you an example, after the introduction of the vaccine, you can see how the death rate is dropping. And that is because your adaptive immune system has been trained and protects you. How long is the protection? I think the best is illustrated in this example. So what people did, they looked at the blood of vaccinated patients and asked how much antibodies are still there so many years after the vaccination. And I apologize, it's pretty small. I need my glasses myself. But the half-life of vaccinia, measles, mumps, rubella, 90 years, 3,000 years, 500 years, 100 years. So the B cells which have been educated in your childhood, protecting you longer than you live. Of course, it's not all. You have here, for example, tetanus, which a half-life of 10 years, which is the reason why people recommend that you get a booster every 10 years to keep the protection up. So that is a memory feature of the adaptive immune system. And looking at this feature, I think it's pretty clear that we can sum up in the strategies of the immune system. So the innate immune system has large numbers, right? As I said, half of the immune cells in your blood are neutrophils. They're almost everywhere, all over your body, ready to fight. And if something happens, they fight immediately, right? We had that in this video, right? We have a lot of these neutrophils. They're all over the body. They're always in close proximity, whatever tissue happens, and they respond. Within less than an hour, they're fighting for your life. And that's the point, right? The goal of the innate immune system is you stay alive. And they're doing good. Otherwise, you wouldn't be here. In contrast, the adaptive immune system has very highly specialized cells, very few of a single specificity. So if these are the foot soldiers, these are the highly trained ninjas. And they need a lot of education. And that's the reason why they need so time to first know what they need to do. We come back to that. But also to expand, right? You start with a handful of cells. They can't do a lot in your body. They need to proliferate and expand. And that takes, of course, days for the cells to be enough numbers that they can actually help you. Which begs the question, of course, why do we bother having an adaptive immune system? And I think it's best illustrated in this cartoon. So if you think back the last time you have been really sick, would you be able to outrun Tom? But the point is, even if the pathogen would not make you kill you directly, if you feel so sick that you can't fend for yourself, that you can't outrun your competitors, you become lunch yourself. If you're not able to find the food you need, don't find shelter. So even then, the pathogen kills you indirectly. So, and there, of course, memory helps. The goal of the adaptive immune system is don't get sick the next time. We have the start of the winter. You will have a cold. And during the winter, the cold will come back. The virus will infect you again. But the second time, you will not even feel that you got infected. Because the adaptive immune system has been trained, knows what to do, and protects you so that you do not get sick and that you're able to outrun Tom. Hopefully, you don't need to do that, but that's a strategy behind it. So we introduce the cells, the features, and the overarching strategy. So, but how do we get there? How do we get, all right, you don't want to have no immune system at all. That would be bad. And you want to have, under resting conditions, an immune system which is calm and collected. You only want the immune system to get activated, sorry, wrong direction, once something happens, like an infection. So how do you get from here to here? And I think one way I would like to illustrate that is with maybe a bit of an odd question. If you try to think for a minute, how would you build an immune system on your own? We don't need to go into details because I want to highlight strategies. So if you think back to your favorite spy movie, war book, thriller, how did they, in this book, in this movie, detect the enemy? How do you know it's a spy? You usually can't go up and ask, are you a spy? Oh yeah, sure, put me to prison. Probably not, right? The enemy will try to hide. Are you able to detect the enemy? And even if you detect it, how do you know it is an enemy? In the Second World War, the Dutch resistance used a password which apparently was very hard to pronounce for non-Dutch speakers. So even if a German soldier would get to know the password, by pronouncing it wrong, they still knew that it was an enemy. So how do you do it? How do you find the approaching airplanes, right? The first time airplanes were novel technology, that's how they tried to detect the incoming airplanes. And once you figured out what is the enemy, how do you fight it efficiently? Right? If you have a burglar at the window trying to break in, of course you could ask an airstrike to bomb the neighborhood, but it's a bit excessive. You want to limit the collateral damage. If you think back for the COVID-19 pandemic, the patients which actually died from SARS-CoV-2 did not die because of the damage of the virus. They died because of an overreacting immune system. You want to avoid collateral damage. And of course, the details of the mechanisms might be different, but whatever strategy you can think of, what is unique, what is different? How do you test it? How you fight the enemy? All these strategies are used by the immune system as well. So let's look at some of the details. So we mentioned that innate immune cells have these receptors given by evolution, which allows them to sense pathogens. And the reason why it works for 600 million years with these receptors is because they recognize bits and pieces from these pathogens that the pathogen cannot just change. Right? A pathogen can evolve to change some proteins, but they can't change DNA, RNA, some essential parts of the cell membrane, like lipopolysaccharide LPS. And then these receptors get activated, the immune cells get activated. So we mentioned that B cells can do that too, but we mentioned also that they're slow, so that comes later. So these receptors activate the innate immune cells. What does it mean? Right? Again, think about the war situation. Your duty is to protect the border wall, the fence next to the enemy. And you make your daily walk and you see a hole in the fence. And you say, okay, and make a note, and then you continue walking. Probably not, right? You running back to your hut, calling your superiors, and make some noise. Back in the days, if you were at the fence of your castle and you saw the enemies approaching, you made literally noise, right? Bells, whistles, whatever, to get your friends and colleagues to fight in. The innate immune system does that with cytokines, which are basically hormones of the immune system. And if you go back to this picture, you can see that the pathogen is here, but the immune cells in the bloodstream hear about it. Of course, it's not really a sound. It's cytokines traveling from here to here, which leads to changes here, which highlight that there's something going on. And then you can see all these brothers and your colleagues swarming in, helping you to fight the enemy. One thing I would like to highlight here is that, of course, different immune cells will respond in different manner. And I think it's nicely illustrated in this video, where in the middle, there was a laser damage. So they shined a high laser onto this tissue, damaging the cells. And these things are recognized by the immune system too. Because in the end, if you think about you have a worm under your skin, chawing its way through your tissue, that is damage, and the immune system needs to respond to that as well. So here, here you can see in red now the neutrophils, and you can see how the neutrophils swarm again to the area which is damaged. And in green here are monocytes, another innate immune cell, which is a bit slower. So they find themselves in the same environment, the same signals, but because cells are different, cells have different receptors and different concentration of those receptors, they will behave in a different fashion. And that is, of course, important to sharing of labor, as we will talk in a second. Okay, so you manage to get the innate immune system activated. But that is only one arm of the immune system. You want to bring in the other arm as well. So for that, you need to activate T cells. So the innate immune cells can present bits and pieces to the T cells to educate them. And for that, I need to explain a little bit more about that. So I mentioned that there's a specialized molecule that presents the antigen for the T cells. And I think the best illustration I've found to explain that is this cartoon. So you have here an antigen-presenting cell. It takes a protein, for example, from a virus. It chops it into pieces and puts these pieces on a plate. And then this plate is presented on the surface for T cells to come and check it out. How does it look like? Oh yeah, by the way, you can see if you have any artistic inclinations, science needs you. This is from a PhD defense of Greek rights. So go ahead. So how does it look? The T cells recognizing these bits and pieces. So that's again a video from the lymph node. You see in green the T cells. That is a bloodstream. That's a lot of T cells, of course. And you can see here in the lymph node the T cells again swarming around. And in red are antigen-presenting cells. And you will see, for example, here how T cells bump into the antigen-presenting cell. And they're basically checking out the plate. Is there something I find interesting? For most T cells, there will never be anything of interest. But if they find something, they get excited. And then you have something like that. The T cell found something on this antigen-presenting cells that the T cell receptor is recognizing. And now they have this hour-long interaction. It's really a deep conversation where they talk about each other, exchange information, and the antigen-presenting cell basically tells the T cell, look, that happened, and we need now this and this and this. And now we have the adaptive side in the party, but then one essential part is still missing. Because you have to compute all these information, and please think about this computing like a black box in your computer. But that comes out is, of course, a pathogen-targeted response. And that makes sense. It's important because if you think about it, a bacteria which is inside of your cell needs a different response than one which is outside. A virus will need a different response than a helminth. If you, again, thinking about the fence at the border, if you go back to your hut and you scream to all your colleagues, the enemy is here, and then they take the gun and run wildly around, that will be not efficient. You want them to go to the fence and follow the tracks, not run around wildly. If there's one guy coming through the fence, that's a different response than if there's a huge gaping hole with one tank after the other, right? You have to adjust your response to the target. And of course, it's very complicated if you look at the details, but in broad terms, we distinguish 3 types of immune responses, which for historical reasons are called type 1, type 2, and type 3. And the weird naming comes from the way that was discovered when they looked at the response of T cells and distinguished that there are different types, different flavors of T cells, which they call Th1, Th2, and Th17. So how do they come along? So I mentioned that there are T cells which express a T cell receptor, but of course, the more you zoom in, the more complicated it gets. And one piece you need to understand additionally is that there are broadly 2 flavors of T cells. One which express CD4 on the surface, one that express CD8. It's just a protein. CD4, CD8 doesn't mean anything. It's just like a house number so that we can remember what is what. The important thing is that CD4 T cells help to orchestrate the immune system, right? So somebody screams, there's the enemy. And then somebody has to tell, okay, you go there, you do this, you do that. To make the target response focused and efficient, that is where the helper T cells come in. The CD8 T cells are the ones which kill. I come back to that in a minute. So let's focus on these helper T cells. How do they know? What? We mentioned that in this interaction, the antigen-presenting cell educates the T cell on what to do. And based on this interaction, the T cells become educated, becoming these different flavors, Th1, Th2, Th17. And how does the antigen-presenting cell know? Well, that comes back to these receptors. So over evolutionary time, these receptors were connected to a particular flavor of response. It worked 60 million years ago. It probably works tomorrow. And this information is transferred then to the T cell. And based on this education, the T cell will become different flavors and having different functions. So how does it look in detail? So we have now these 3 responses. And let's think, for example, about extracellular bacteria first, because that's the easiest to understand and imagine. So these are cells which are able to attack these bacteria, these fungi directly. And the most easy way to understand is here a macrophage recognizing these receptors, the bacteria, and just gulping them up. Huh? Gone. So after all these bacteria are ingested, that is this part, they're digested, like in your stomach. So what these macrophages have are these vesicles which contain a lot of nasty stuff, which is dumped onto the bacteria. If you think at your bleach you use at home, you can find this bleach in these vesicles as well. And that, of course, kills them. One strategy, if you want to fish, you can, of course, pick a fish one at a time. A different strategy would be to use a net. And as I said, whatever you can think of, the immune system thought of it too. The immune system have a net too. That is an example of a neutrophil where you can see the DNA spilling out to form a net. So what these cells do, they take the DNA, loosen it up, mix it with all the toxic stuff they have inside of them, and dump it onto bacteria, or in this case, on cells. Like the net when you fish is to collect and, with all the toxins, to kill. How does it look? You have your neutrophils. In red is DNA outside. And you can see, once I get activated, how this DNA is spilling out. Pus, so this yellowish liquid, if you have an infection, is literally dead neutrophils and these nets. So, if you have extracellular stuff, which is small enough that you can digest it or to catch it, that's a strategy to do. But what do you do if the stuff is too big? Right? A worm under your skin might be 1,000,000 times bigger than any immune cell. So the strategy here of these cells, which have all these different vesicles with all the different toxins, you can't ingest it and then digest it. The strategy has to change. So what they do instead of dumping all these toxins inside of the cell on the bacteria, they're now dumping all these toxins outside on the surface of the cell onto the worm, right? If you're thinking about the hole in the fence and there's a tank coming in, your net will not do much. That might work with 1 or 2 soldiers, but not with a tank. So with a tank, you're trying to punch a hole into the tank, for example, with a grenade. That's a strategy here, to try to damage the worm from the outside to kill it that way. And last but not least, what do you do if the pathogen is inside? It's out of reach. You can't directly attack the virus, the bacteria, if it's inside the cell. Here's the strategy is, we have so many cells, better safe than sorry. We kill the cell, right? The virus is more dangerous than the loss of a few cells or a few thousand. So you have here now cells that are able to kill other cells. NK cells, cytotoxic T cells, macrophages. And to give you an example how that looks, that is from an NK cell. You can see a tumor cell. The same would work with a virus-infected cell. The NK cell comes and kills the tumor cell. So the NK cell comes in, connects, gives a kiss of death, and then the cell is dying. Whether it's a tumor cell or a virus-infected cell, the response would be the same. And one thing I would like to highlight here, if you look at the response of these cells, they shrink, and you see this blepping. We come back to that in a second. The important thing is that there is a distinction now between your war movie and the immune system. In the movie, you see the people shooting around, and then there's blood spilling around everywhere. It's pretty messy, right? Usually the people which do the shooting are not the ones which clean up afterwards. In the case of immune system, that does happen, right? These macrophages that clean up are the ones which are involved in the action as well. So the strategy here of the immune system is a little bit different. And I think it's more similar to what Garfield is experiencing here. So Garfield learns from his owner, John, that plants respond to words. And Garfield, as a good scientist, does an experiment and tells the plant to die. And the plant does. And in the end, that is the strategy the immune system is using, right? Remind you, the immune system does not want all this messy stuff spilled around. What it wants is that the cell is killing itself and putting itself in bits and pieces, which can be easily brought to the garbage dump, which in this case are macrophages cleaning up these bits and pieces. This blabbing you saw in the video, I think I have it here once again. After the kiss of death, this blabbing is preparing the cell to be eaten by other immune cells. And this is an active process. The cell has to make an effort. It's a suicide. And I think nicely illustrated by caspase-3, which is a part of this active process. Doesn't matter what it is, but you will see it here light up in a second in red. So you have here a tumor cell that will be in white and a NK cell giving the kiss of death. And then the tumor cells will actively kill itself. So the NK cell comes in, the kiss of death, and then the tumor cells or a virus-infected cell will kill itself. So with this, we have the three parts, right? How do you protect your body against bacteria outside, inside, and things which are too big? And with this, I would like to come back to our initial question, right? Why are you still alive? Of course, the reason why is the immune system. Your immune system is highly efficient at eliminating microbes. If you're in the garden barefoot and you step on a rusty nail, there will be millions, thousands of different species of bacteria and viruses entering your body. But by all likelihood, you will not get sick at all. Because the immune system is highly efficient at cleaning up the mess. Literally, the bacteria and the tissue damage is all cleaned up by the immune system. So only a tiny fraction of these organisms can make us sick. And these are organisms that have learned to avoid parts of the immune system. You cannot understand why the antelopes are so fast if you don't understand the cheetah and vice versa. So it's a co-evolution between the immune system and the pathogens. And that gets us from here to here. Of course, one essential part is still missing. You don't want to stay here. You don't want to have a car without a brake. That will give disaster. You need to go back to a resting state to reach homeostasis. Because one of the things a lot of people miss is that the immune system is actually dangerous. The wrong immune response under the wrong setting is bad. So the same immune response can be good or bad, depending on the situation. So if you go on holiday and you eat a fruit you never had before, you don't want your immune system to respond to that, right? You want the immune system to tolerate it so that you can enjoy it the next holiday again. But if it's a tumor cell and your immune system is tolerating it, that's disaster. And similar, if you have an infection, you want the immune system to get activated. But if it's not targeting a virus, but rather it targets a piece of you, your tissue, or something harmless like pollen in the air, that leads to destruction. Every single autoimmune disease is because of the adaptive immune response targeting the wrong thing. And so with that, I would like to finish. I introduced the cells. Remember, T and B, that's adaptive. Everything else is easy. The features and the strategies. And hopefully that helps. And if there are any questions,